friend of this club for many years. John Plischke is actually a founding member of the Western PA Mushroom Club and is the recipient of 2 of NAMA's top awards. He is the author of Morel Mushrooms and Their Poisonous Lookalikes, and a book called Good Mushroom, Bad Mushroom, which is actually a very cool book. He currently serves as an expert on the National Poison Control Hotline, and I am very pleased to introduce to you the speaker, John Plischke.
So today we're going to take a little bit of a look on how we send our mushrooms in for DNA testing, or if you just see a mushroom that gets DNA testing, like on Facebook, people are posting them all the time, and what sort of goes on behind the scenes. A lot of people tell me that they send a mushroom in, it takes a year, it takes 2 years, takes whatever, and they don't know why it takes so long, and you're going to see a little bit of the process and, you know, find out how it works. And there's 3 different labs that are doing DNA testing pretty much now, and they all do it very similar in the style. So in order to keep track of our mushrooms, we use iNaturalist. Some people use Mushroom Observer, and we put our pictures on there, and we keep track of the mushrooms, the photos, and our DNA. Currently on iNaturalist, there are over 95,000 mushrooms that have been DNA tested. We think that within the next few years, it'll be 2-3 years, it might be 200,000. It's going to exponentially up every year. It's becoming commonplace. When I started out mushrooming, maybe a scientist took a DNA test. Now I meet just regular people that are out doing it, having fun. They don't have to know many mushrooms at all. So back in NAMA 2005 in Vermont, we did that was our largest scale mushroom DNA testing at a Foray we ever did, and we tested 1,700 mushrooms. That's like tons of the collections. We were up to 3-4 o'clock in the morning every night, putting little pieces of mushroom in the dehydrator and doing it there. This year at NAMA, Hartsinger will be taking in charge in Vermont, and his goal when we had a meeting is to DNA test every single mushroom that comes in at the Foray. I don't know if we're going to be able to do it, but we're going to try. At least we have to beat the 1,700 record. It's going to be a tough thing. So if you're coming to NAMA, your stuff, hopefully a lot of it gets DNA tested.
Are they testing in Vermont Foray?
NAMA in Vermont, and we'll be doing it in NEF too. NEF, they said they're going to do at least 1,000 is what their goal is. So this was last year at NAMA in Vermont, where we did 1,700 mushrooms and we DNA tested them. And Ariel, the lady in the middle there, got our picture taken. It's me and Alan Rockefeller and Ariel. Together we identified over 1 million mushrooms on iNaturalist.
Wow.
And so the funny question is, people ask me, well, how many mushrooms did the 3 of you identify at NAMA, if you did 1 million mushrooms on iNaturalist? And the answer is together, working the whole Foray, the 3 of us identified 0 mushrooms. I was busy, doing the 1,700 DNAs. I was up to 4 o'clock every morning, and I just didn't have time to put a name on a single one. So Garrett took this picture. I think I'm in the back where I was there. This was 4 o'clock in the morning at the NAMA. Everybody's in bed, and that's what time I went to bed. He took a picture as we were leaving. So now DNA is becoming, and local clubs are involved. Some local clubs more than others, and a lot of local clubs are becoming involved. And I was at the West Virginia Mushroom Club Foray this year. And West Virginia Mushroom Club is like an active club like this club up here. They have 1 Foray a year. It's 3 days long. They do not have any walks. They don't have any talks. They don't have a newsletter. Just once a year they get together at a Blackwater Falls State Park in West Virginia Resort, and they collect mushrooms. They said, we're going to do some DNA this year. And, you don't have high hopes. People aren't used to doing it. They're not trained. And Shannon Nix gave a talk and showed people how to use iNaturalist. And at this Foray, we had I think it was 84 people take iNaturalist pictures. That's a huge number of people doing it at a Foray. And they took over 1,000 DNA samples at the Foray. For a club that doesn't have any meetings or any walks, it's amazing, you know? How's that happen? I'm asking myself, at a Foray, have I done it like a regular club Foray? I've never done 1,000 mushrooms. You know, how did it happen in West Virginia? But I think it's fun disc, and people are getting people involved, and they read about the Rare Fungi Challenge, and they want to find out what they have. But this is, you know, West Virginia Club Foray this year. It's an INAT project. I haven't even added my stuff to the project yet. I think I sequenced about 200 or more there, at least. And my pictures aren't on there. That's their result, their collections, 1,200 photos. So if you want to take part in DNA for free, you don't have to be a scientist. You don't have to know anything about it. They have 3 dates every year that you can take part in one of the DNA projects. This fall, it's coming up October 17th to 26th. And you join the INAT project. You take a picture with the app on your phone. You don't have to have a good camera or anything like that. You can use a better camera. And you send your mushrooms in for DNA testing. You know, in the past, it's been you send 10 mushrooms in for free. And then every year, then if you want more than 10, you pay $3 a mushroom, which is below cost. You know, I think he's subsidizing that somewhat.
Yeah, he's asking for $3.
Yeah, he asked for $3.
Yeah.
So what we do is we photograph the mushrooms in the woods. You know, we use our cell phones. I also use I use a big digital SLR. So I take my cell phone picture to geotag the location. Then I'll take a nicer picture with my good camera. And later on when I get home, I'll add the good picture to the cell phone picture. But you don't have to. I know other people that stack photos, so it's real involved. But as long as you get a picture up there, that's all that really matters. So that's what we do in the field. We take a picture of the mushroom. We take a picture of a slip with a unique number. You know, unique number. You download the slips off Steve's website for the Myco Blitz, and once it goes on iNaturalist, you know, go to the lab, and eventually he'll put the DNA sequence on your mushroom and tell you about it. So what I do when I collect the mushrooms in the woods, I'll take the mushroom and I'll wrap it on the ID slip. I'll fold it up, just because all I'm taking is a sample. I don't take the whole mushroom. I just take a little piece. It could be as small as a grain of rice. Usually I break up a ½ piece or something. Wrap it in the slip, fold it up. When I get back to my house, or if I'm camping, I'll put it in the dehydrator at my tent site. I'll dry all my mushrooms at night. Set the dryer at about 120 degrees, and by morning they're usually dry. This is how my trays look. In the old days, like here's the tray, I used to have big pieces of the mushroom. You know, we thought it was important to stick in herbariums, and it still is important to do. But now the labs don't want big pieces like this. I take little pieces, put them in a bottle cap so they don't blow around. Sometimes I use stickers, or I put the slips on them. And that's how I dry the mushrooms. And you say, you know, people will say, how come we don't take these big collections? Most herbarium collections might have, you know, 10 big full-size amanitas. You gotta realize that when Steve saves these little vouchers, he has 70,000 mushrooms. You know, he used to store them in his house. How much space do you think, you know, big gallon Ziploc bag, if you had 70,000 of them? I don't even know if you have room to live or walk around your house, you know? I don't know how he stores these mushrooms. You know, how he finds the space. So Steve always says the best results for sequencing your mushrooms is to stick them in the dehydrator as soon as you pick them. If you can't put them in the dehydrator, if you're busy, you come home from work, you know, you're mushroom hunting, to put them in the refrigerator the next day. In the refrigerator, you won't lose much quality on the sequencing results, but get them dried faster and you get better results.
What about sun-dried?
I think sun-dried will work. You probably just have less quality of results. I think the faster you dry them, you know, the better. If they're slow, it doesn't work as good for DNA. You might have a 50% success rate or something less.
You also want to watch out for temperatures that are too high also, right?
Yeah, I think if you get way too high can hurt, because I usually use, some people like 105, some people tell me 10. I usually use about 115, 120. And you go a little higher. We had a food microblitz that NAMA sponsored it. We went to the grocery store and bought, like Chinese grocery store, and bought all these boll leaves to see if they were really Boletus edulis. And we tested them to see what they were. And surprisingly, like 90% of the dried mushrooms failed testing. And we're thinking that they might have been dried like at 190, you know, some extreme temperature that cooked the DNA out of them. So you probably can get too high.
Somebody told me once that they dehydrate mushrooms often, the commercially dehydrated ones are dehydrated in a microwave, which probably is gonna ruin.
The microwave probably ruin them.
Yeah.
So we take our mushrooms out of the dehydrator, I store them in Ziploc bags. I recommend using snack size Ziploc bags. Steve will take snack size or sandwich size. The Ohio DNA lab only takes snack size, but all the labs take snack size. So if you buy that one size bag, you're never get in trouble no matter which lab you use. So snack size is a good size. The blue bag I think is at NAMA last year when we're doing some, yeah, it was NAMA and I had 1,700 mushrooms in that bag.
Wow.
That's the picture on the left is my buddy Garrett's, that's how he stores them at home. So you must add the INAT number to your slip. So if you're out taking a picture in the woods, you get an INAT number, it's the last digits on the web page. And you gotta write that on your slip if you're gonna send it in for DNA. And you ought to take your voucher number right here on your INAT observation. And once you send them in, maybe, you know, a year later, year and a half later, depends how lucky, it could be 6 months. The lab's fast. They'll come by, somebody will come by from the lab, they'll put your DNA sequence, you have a little link you can click to see the blast results. There's a thing called recent consensus. If it's higher than 100, that means it's really good. And that has 300, so that's a great read. You might have other things that they put. And usually they also put notes on your mushrooms when you get them back. They might say that this is the first mushroom ever sequenced of that species. You know, there's no other records of it. It might be a rare one. There might only be 3 mushrooms in the whole world that ever, you know, they tell you neat stuff about your mushroom when you find a lot of them. It often happens. So there's custom fields that you can create, like Hart likes to create a sequencing request field. So now Hart says that if you send in the fundisk or doing that, there's a box where you can put request additional, you know, redo the sequence if it failed. So you can request it to be done again now in iNaturalist. Before, you know, it wasn't what you could do, but that's a new field. Once your specimen's all dry hydrated, you put them in a box, you know, box them up, send them to one of the 3 labs for DNA processing. The labs right now that you can use that are affordable is Makota Lab, I tell you they're $3. The Ohio Mushroom DNA Lab, which is this one here. They're currently shut down, but they should be opening back up hopefully next year. They're free, they're 100% free. You know, they said now they have a backlog of like 8,000 mushrooms they have to test. And as soon as they get those ones done, they're gonna open it up for people to send in again. Everybody's always getting backlog. You know, when you say send our mushrooms for free, and, you know, people just send boxes and boxes and boxes. And then now there's one called, the Hart opened up his own sequencing lab. He charges $5 a mushroom. And Hart's lab, though, I think you have to validate them yourself. All the other 2 labs validate them for you. And so this is what I typically do now. I go to Walmart, I buy these little shoe boxes. And these are 99 cents a piece. You know, buy 10 of them at a time for 10 bucks. And when I take my mushrooms out of the dehydrator, I organize my Ziploc bags by alphabetical, you know, order for the slips. And I put the box in a cardboard box, mail them off to the lab. So they're already organized, ready to go. It's pretty easy to do. Store them at my house that way if I'm working on them. So all the labs tend to have a lot of the same equipment. They might do it a little differently, but let's look at, and you can do it yourself too at home. Let's look at how they do it. This is my old home lab in my bedroom. I was using this maybe 3 months ago. And I got my thermocycler.
You sleep in that chair?
Thermocycler right there. Another thermocycler, one in the back. I got like centrifuges here. This is a medical freezer. You don't have to use one, but it's minus 20 degrees. It keeps my specimens good, my chemicals in there good, and specimens. I have little racks up here that I use. I got a table that I do most of my chemical work on. Right there's my stainless. I do some on the other table. Got a few more thermos, I got bags over here, slips. And it got a little bit crowded in there, especially if, you know, me and Garrett, we're working, he comes over and we work on DNAs together. There's my bookshelf with DNA equipment. My filing cabinet, I keep specimens in the filing cabinet. It can be done in a small home lab, you don't need a lot. I know Sigrid from the New York City Club, she has a couple little boxes right on her kitchen table, you know, her kitchen counter. You don't need a lot of space. Then I figured out, you know, using my home lab, I had some extra lines running. It wasn't quite enough space, and we were doing like 1,000 mushrooms, so we carried thermocyclers down and we put them on the kitchen table, we put them on the kitchen countertop, and my wife's like, man, can you guys get this stuff? You're in my way. You know, and we were doing stuff in there. Gary and I set up 2 days and we did like 1,000 mushrooms. We thermocycled, got them all done.
Wow.
Takes a little bit of time. So, you know, things are always being done in the lab, whether it's working on runs, getting better equipment. There's always backlogs. You know, my backlog now is moving it to the basement. You know, I always hope that I'll be better organized, it'll be quicker, but I think now it's just gonna make me more work. Because if I have a bigger lab, that means instead of doing 2,000 or 3,000 mushrooms at home, maybe I wanna do 4 or 5, you just wanna do more and more and more, and it makes it worse rather than better. I don't know, we'll find out. Here's my new lab, starting out in the basement. So I got, you know, trying to collect lab equipment, and stuff's expensive. These are actual lab tables, they probably weigh 600 pounds a piece. When I bought them, I hurt my finger and I could barely lift. I had to hire movers to get them in the house. And I got these at University of Pittsburgh surplus. I think I paid $120 for a lab table. You know, if you were to buy a new one, it's probably $5,000. So I got a lab table here, got one here. This is on my TV here, and this official lab table. This is an old lab table from Ohio Science School that I have my desk at. And there's another table on the other side. I have thermocyclers. Thermocyclers up here, I think I have 7 thermocyclers there. All these boxes under here are PCR tips for doing the PCR work. They're like $700 a box if you order them from Thermo Fisher. But you can get stuff cheap, you know, it's not, you think buying lab equipment's unaffordable. I went and got them at surplus, and I paid $10 a piece for the boxes.
Where do you get the thermocyclers, on eBay or?
On eBay,
Yeah.
Facebook Marketplace, but usually eBay is where I buy most of them. I've been frustrated lately, I've had bad luck with them. I would say half the thermocycles I buy don't work. But you know, I might pay between $50 and $400 for most of the thermocyclers I have. I have one I paid, 2 I paid a little bit more for. But you know, if you pay $100, every other one works. $200 isn't that bad compared to $12,000 for a new one. You know, I'm going through a testing phase. I still gotta test some thermocyclers. That's my basement lab. You see I've got lab cabinets there. Here's a view from the other end. You can see my other table. Got different supplies under these lab cabinets and cabinets on the wall. These lab cabinets, I just bought those in the back. Kim's been getting a little, Kim's getting tired with me because I say, I found some lab cabinets. They're 5 hours away from home. We gotta go get them. And you know, I might get them for $50 for the lab cabinet or $100. And if I look on the line, it's $1,000. So you know, I have to drive a few hours, pick it up, and I find them all on Facebook Marketplace, you know, for a reasonable amount of money. Some more of my lab stuff. These are, right here is a lot of the specimens I got. There's probably about 100 specimens in each of these boxes. And I have a couple more places where I have them stored. I didn't carry them all downstairs yet. These are the new lab cabinets I got in the back. Started to put specimens in here too. Another view of the lab from the other end. So, get a lot of stuff off eBay. This is a new centrifuge I bought. I got 2. I had one, and I figured if we're gonna do like 1,000 mushrooms in 2 days, I need 2 because they only hold a limited number. And at Fisher Scientific, this thermocycler is $1,200. And I got it on eBay for 152. So, you know, it's affordable. It's expensive, but, you know, people pay that much for a by the time you buy a good microscope, you can have a DNA lab set up at your house for the same price. There's a Fisher, actually it's more, $1,570 a Fisher. And mine was brand new too. But it's other equipment there. There's a thermocycler I just bought by this Eppendorf on eBay for $1,200. That's my most expensive one. It was brand new. I looked it up before I bid on it. And at the scientific stores, they went, I think $1,300. $13,000 for it. So I gotta test this, hopefully this one works and I won't have any problems with it.
What's a thermocycler?
It, you put your tubes in, it heats and replicates the DNA. Here's one I bought, with shipping it was $126, but I think I bought that thermocycler there for $15. So that was you know, that's affordable. Wasn't as good name brand as the other ones, but. So at a DNA lab, once you set your stuff, I showed a little at my lab, how we process stuff. You're gonna send your mushrooms off to the DNA lab. And eventually they get to Brian Hunt. He's the first person that's gonna work on your mushrooms. And that's Brian, he's sitting in his house, this is his house in Indiana. And he has a little light box here. And he takes a little he takes a tweezer, and he takes your mushroom and he breaks off the smallest piece he can see. You know, this grain of rice is way, way too big. It's like a little fleck that you have to put on your reading glasses and look down. That's all the bigger you want to use now. And he puts it in the tube, a little plastic tube. He writes like iNaturalist number down on the chart, and he does all that. He prepares your mushrooms. Here's the tubes he uses right here. A box of these tubes for 1,000 of them is like $40. And there's this little homemade light box, and these little racks that he puts them in. And so if you're taking part in the microblitz, you say, what takes so long? Steve gets the mushroom. You mail Steve to the lab there. He gets them. He has to wait before Brian lives in Indiana. Steve's in Michigan now, you know, maybe 5 hours apart. Steve doesn't drive 10 mushrooms down to Indiana. He's gonna wait till he fills up the back seat of his SUV, the trunk, everything's piled to the ceiling. You know, he's bringing 20,000 samples down to Brian, or 10,000, whatever he can fill up his vehicle with. So it might take him a month, you know, if it's during the middle of the micro blitz, maybe he has enough to drive a load right away. But maybe he doesn't. Maybe it takes him 6 months before he gets enough to drive down to Brian's. You know, I can't tell you exact times, I'm sure it differs, but that's a big delay. Then it gets to Brian in the lab. And Brian says, okay, he posts on Facebook. And he'll tell you he gets the mushrooms, he'll tell you what he's doing, what he's up to. And Brian will say, I got 20,000 mushrooms today. Steve dropped them off. What do you think it takes Brian to sit there and take your mushroom, open the Ziploc bag, you know, find a piece that small, put it in the tube, then you gotta hold the tube up to the light to make sure it fit in the tube, and do that 20,000 times, you know? Then once Brian gets all 20,000 of them done, he calls Steve up and says, mushrooms are ready, come pick them up and bring me another load. You know, that takes quite a long period of time. Sometimes it's fast, sometimes it's not. Brian posts on Facebook and YouTube, he'll have little videos and he'll show you exactly what he does. He'll show you how to take the samples. Tells you all sorts of detailed information. I like to watch how it's all done behind the scenes. And you know, he's getting many thousands at a time. Talked about how he did in the cart. And Brian will post, Steve will post, and I'll say, these boxes just came in, or this box came in. And you can see the boxes. If you're sending mushrooms to the lab, and you want to know when he gets them, I got a secret for you. Draw a little smiley face on your box, put a sticker on it. You know at airports how you put ribbons on your suitcases? Mark your box. And that way, when they get at the lab and they start picking the tissue, you know it's there. You're saying, hey, that's my box of mushrooms. And people are starting to do that, and you know when he gets them, when they're getting worked on. That's a little tip that you can do. So there's your 10,000 mushrooms that Brian's working on. Put some of these little racks, he puts stickers on them. Marking your box. What Brian does is he'll get the mushrooms, here's the Ziploc bags, and put them in order. He prints a little sticker, he puts stickers on the bags. He preps them pretty good. So other people beside Brian that will show you what they do behind the scenes is Jessica. She's on Facebook, she's on YouTube. She's a sequence validator, and she validates sequences for the Ohio Mushroom DNA Lab, and she also validates for Fundisk. And she'll go on Facebook and she'll make a video and she'll teach you how to validate a sequence. She'll show you how you do everything. She teaches classes on it. You know, all this is free to watch. So if you want to learn more about what goes on behind the scenes, you go look at one of Jessica's videos. Her website's called Midwest Fungi Finds on YouTube, so you can watch all that. She tells you lots of stuff. Here she's telling about how they do holotypes. In the old days, I was always taught that, you know, it was always one mushroom. We didn't have hybrid mushrooms. If you said there was a hybrid mushroom, you didn't know what you were talking about, you know. Talk to most of the mycologists, they'll tell you that. But Jessica says now, and a lot of people, they're thinking tons of mushrooms are hybrids. When they test a mushroom, maybe 1 in 20, it's a hybrid, and there's actually 2 sequences, and they have to separate the 2 sequences from the mushroom, and they'll post them both on your observation now. So there are hybrid mushrooms they're finding out. Another one is, this is the guy that runs the Ohio Mushroom DNA Lab. He'll be at the Ohio Mushroom Festival next weekend giving a talk and showing what he does. Scott Osmond's, Scott does a lot of validating for Fundisk and for the Ohio Lab. He has a good bit of videos, and you can watch how he does it. Steve, he'll post on, he doesn't have a YouTube, but he'll post on Facebook. Sometimes he gives little classes. He'll show how Steve does the validating and what happens to your specimens. Like if you join some groups like fungi sequencing, fungal sequencing results on Facebook, you know, Steve might post that tomorrow he's going to do a talk. I'll put the Zoom link on and put the password. Just click on the link and watch his talk. I do that a good bit. So there's one of the last talks I went to that he did, sequence validation discussion. He showed how he did it. And I watched Steve do the sequence validating. I watched Jessica and a number of other people, and they all do it differently, but they all do it pretty good. And there's always something to learn no matter who's doing it. Another one of Steve's talks. I think a couple days ago, Jessica and Hart did a thing on DNA sequencing at Summit Metro Parks, and next weekend I'll be at Jessica and Cal will be talking at the Ohio Mushroom Festival on DNA. There's a lot of DNA programs that are going on all over the place. Brian does his things on YouTube too. This is PCR extraction. He shows you how to, you know, pick the mushrooms like I talked about. Tells us detailed instructions how he does it. He also uses a little printer, and he prints these little barcodes puts it on the individual bags, and he puts them on the racks. And once he gets your mushroom, they'll download the Phalanophyte Naturalist, and the little code is to your picture. So he can scan the little code on there, and the picture of your mushroom when he's working on it comes up right from iNaturalist, and he can look at it, because sometimes you get a log and maybe there's a brown speck or a gray speck. You can see what you want him to sequence. So Brian says it takes him, he can do about a mushroom a minute doing it. I try to do it, and to do a 96 well plate, it takes me about 3 hours. I'm not nearly as fast as Brian, you know, but it's a hard thing to do. And you can do this at home too yourself. There's a little chart you download. I told you how you take a piece. Here's the chart that you use. You just have to put the position in the tray. You put your INAT number, your voucher number, INAT number, and the position in the tray is on there. And once you do that, you know, it's done. So if you want your mushrooms to get done really, really fast and get more experienced at it, you watch a few videos, you can buy the little plastic tubes, 1,000 for $40. You can tissue sample at home. You can mail them to the lab in tubes. And instead of having to wait for all this to get done, yours will probably be priority on the lines, because when they get have to do a nanopore run, these little cartridges, they expire in like a month or something like that. They go bad. So if he has to buy like 10 of them, you know, $250 a piece, and they're going to go bad, he's gonna get everything that's ready and do those first.
John, how do you label the tubes?
The tubes?
Yeah,
So the tubes we write on the very top of the tube is clear. We write the position on the rack.
no, but I mean somebody who's submitting material in tubes.
Yeah, we submit them in those little plastic racks, and we'll put what position in the rack, like if it's one, or like what number, 1 to 12.
So you fill out, it's like a sheet you fill
Yeah, the
out.
sheet, like, let me go
Oh, I
back.
see.
Right,
Okay.
right, oh, sorry. Right there is the sheet we fill out.
Oh, okay.
And then
that's
we
what I
write-
would fill out if I submitted?
Yeah, if you submit them tubes, you fill out that little sheet that says your INAT, or your mushroom observer number, your voucher number, and there's little A1 is the first position of the rack, first hole.
Okay, I see.
And then you might want to label, like on this is ones I submitted, this is plate 3 that I submitted, so I might write plate 3 on it.
Yeah, this is an improvement, because Fundus a few years ago wanted you to tape.
Yeah, we don't tape anything to
a
them.
Mushroom observer number or something to the.
Yeah, we don't do any of that, we
Okay,
just,
This is an improvement.
it's way faster, way easier. I did 5 plates of mushrooms in 2 days. It took me 2 days to pick them. You know, me and Garrett Teller, we picked them, we ran the machines, we did all that. And 3 of the plates came back perfect, you know, a 98% success ratio. I had 2 plates that failed. I think I had a bad thermocycle, you know, I bought a bad one. So I gotta rerun 2 plates, and eventually I'll figure out which thermocyclers work and which ones to throw in the trash. It's frustrating a little bit, but it's pretty simple to do.
Don't throw them in the trash, sell them, resell them on eBay.
No, I don't wanna rip anybody, they're gone.
You get a bad reading.
Alright, so here's how we'll do it. Let me go back one. So to start it out, Steve has a thing on Protocols.io, it's a website. And it tells you all the protocols the lab wrote out. He wrote detailed instructions. So you can download how to do all these steps. Once you download it, I have a little lab book. You can see this is how we label the steps. This is plate number 3, so it has 3 across. I mean, that's the 3rd plate we did. And this is 1, 2, 3, we just number 1 through 12. That's all we do. And fill out the chart. So that's all you have to mark on your tubes. That's how I mark mine. This is Garrett and I in the lab. So we got our specimens, we picked them, we put them in the tubes. Now we're gonna use a pipette. I'm gonna put some liquid in them, get them ready to PCR. We use XAMP. We use 20 UL of it into the tube. That's all you do, is take a pipette, 20 UL into the tube. Once the strips, you know, we put this pipette into the strip, we tap it, make sure the mushroom sample's in it. There's no air bubbles. Then we spin them down for 10 minutes in the thermocycler. After that, we're at 50 milliliters of molecular water to the XAMP. We don't have to put new tips on because we're not touching anything in this step. We use an 8-channel pipette. The pipette, if you're buying one, you don't have to do this part at home, but if you wanted to do it, the pipette is about $40 used on eBay. So it's not that much. If you bought a new one, my friend Garrett, he went to buy a new digital one. He said, I'm gonna get a new digital one better than you. I think he spent $1,200 on the pipette. And me and him were in the lab working. I got mine out, works perfectly. I'm going, he's getting this digital. It's like, damn thing don't work. You're gonna get it set, couldn't get it adjusted.
Oh, no.
Wasted $1,200 on a pipette. My $40 one works great. So then, we do the PCR for the first time. We have to run the PCRs twice. This one I'm using the PCR I paid $15 for. Took, what's the time on there? It says 40 minutes. We'll have several in one. We set the PCR to 60 minutes at 80 degrees, then hold it at 4 degrees, indefinite hold that cools them. After we prep them in the PCR that way, if we're not gonna do anything else, put them in the -20 freezer. Or if we have time, we might, you know, leave them out of the freezer and start the next step. You see, we had 4 PCRs working that day, doing the first step of them. So once the first PCR is done, you know, put them in the medical freezer. Medical freezer is my new one there cost $1,000, but I have a used one that cost me $200. Or you could use a regular freezer. I know Secret uses their regular freezer. She puts them in a styrofoam container. I think what the difference is, regular freezers often thaw and dethaw, and that causes issues with storing chemicals long-term, and a medical freezer doesn't do that, it's a little bit
So
colder.
a deep freeze chest freezer would be good though.
That would probably be a lot better than one that thawed and dethawed, you know, that was rust-free. So then, we go to the protocols IO, we download Steve's manual, he has it all written down and shares his procedure. And we follow our things, we take our racks out of the freezer, and get ready to start. And what I do now, to save time, you know, I don't have to wait as long, is I buy these plates, and they're chemically tagged, and each one of these plates over here has like a little green colored liquid, and it has a chemical that will bind to the mushroom. And so at the very end of the step, we could dump 1,000 mushrooms all together into one little tube of liquid, and when Steve runs it through the nanopore, he could tell what each mushroom was and what position of tube, where everything was, even though it's all dumped together in a single tube.
How does that work?
And one of these plates, if you buy them off Steve, it's $100 per plate. So instead of being, if you're doing some work yourself, instead of being $3 a mushroom, it's gonna cost you $1 a mushroom. So if you wanna do a lot of DNA sequencing, buy a thermocycle, and now it's costing you $1 a mushroom. So you buy the plate, you don't send any mushrooms to Steve. You just put the little pieces in there, do what we're doing now, and you just send them a couple drops of liquid, and they'll do the rest. So his robot makes these plates. We get them, there's foil on them. Usually I peel the foil off, but you can inject right through the foil. So now peel the foil to remove it. Sort of green on the inside. And it has a plate position, like on the chart I told you we filled out. You can see it's row A through H, and the plate position 1 through 12. And that's what we're recording on our paper chart. So I'm going to use our samples, and the blue rack is what we're going to use. And for DNA application, we take 7 ULs from the top row of the blue rack, and that's what we'll use, and we'll pipette it into the new plate, into the plate we bought from Steve. And the hardest part for me is to see how much liquid gets into these tubes. I have to look across, make sure they're all have the same amount. There's only about that much liquid in the tip of the tube. And then we eject the tips in the trash. So every row we have to eject the tips, put new tips in. So we put it in, we suck it in, suck it down 2 or 3 times to mix it up. Then we throw it in the trash till we get all 12 rows of the plate done. We like Steve's rack's a skirted rack, so Epidorf thermocyclers work better. If you have one that didn't take skirted racks, you have to break the rack off or use a different plate. Then we put in our PCR machine. Here's my settings. It's 94 degrees for 1 minute. It repeats 30 times at 94, 51, 72. And it sounds like a lot of work. Once you put in a machine, you program your machine, just load up the machine, you tell it to run for an hour. You can go off and go out to dinner, and you can do whatever, come back, it'll be fine. So you can leave. It's not like you have to sit here for the whole hour. You can be working on some other mushroom project. So we set the machine, go off and start the next batch. So we label it by the run on the plate. If anything fails, what we do after we put them in Steve's thing is we stick these plates in the freezer, in our medical freezer, and we can pull them out again if the plate failed, and we could theoretically try it again without having to take the tissue samples or anything like that. So we're done, we've got our plate done. What we're gonna do is pull our all our mushroom stuff into the single tube. And usually we keep the cat out of the lab, but the lab cat's allowed in now because we got all the tough stuff done, so you can see the lab cat's right there. Usually Garrett says, "Get the lab cat out of here." But he's allowed in the room. She's allowed in the room. And so we take all our plates and we put our mushrooms into a single tube like this. That tube can hold 1,000 the juice from 1,000 mushrooms that we're going to sequence, that we PCR'd. So everything's dumped in. You're not sending it in. We would have sent 1,000 mushrooms to Steve this way. You don't have to send them 1,000 tubes. You're taking all the stuff from your 1,000 tubes and putting it in one tube and melting in a single tube. Once Steve gets in the lab, you're gonna melt it off, it's gonna come to the lab. And once Steve gets in the lab, he's gonna use 1 drop of that, of your tube, from the 1,000 mushrooms that put in a nanopore. So they come here to Michigan now. Used to go to Steve's house, and Ryan there, he'll get your mushrooms. He's Steve's full-time employee. So Steve's not there and Steve's working, Ryan will be there. This is a little bit of Steve's lab. You can see that's his medical freezer and all the plates that he has. Each plate has 100 mushrooms. Some of the nanopore cells that they use. It's master mix in his lab. A lot of primers that he uses. That's all his plates that he has ready in his lab. These were herbarium. Right now he's trying to do type specimens from the herbarium so we get names on stuff. And these are all the specimens from the Theris Herbarium in California that he has organized, getting ready to test them. Here's a box of mushrooms that was in the lab from Rob Hollick and Hu Fang. That's how many mushrooms Hu Fang collected. I think the big box in the back is Hu Fang's. And she lives in Michigan and she sends him, I think last year she sent Steve 4,000 mushroom samples. I mean, that's dedication. If you're gonna do 4,000 mushroom samples, that might take the majority of all your free weekends to work on. So we had a NAMA food microblitz. That's some of the stuff Steve was working on in the lab. I'm not gonna play these movies, but some of the Steve lab stuff. It's going into his lab. That's his thermocyclers. He used to use an Epidorf, but I guess he got some new ones now. Where was pictures from his lab. That's all the boxes of pipette tips that Steve has. You know, that's a lot of pipette tips in the cases. And that's 2 of his new robots. Like he has robots now. He used to do the pipette and then get in the little trays that we got him, little trays we got to do our DNA in. He used to do all that by hand, pipetting. Now he has a robot that, you know, go, do all the pipetting work and get all the plates ready. And he can tell the robot to do it. Maybe it takes an hour to get it ready. But he's not sitting there, you know, tediously doing one at a time. His robots are doing it. That's 2 new robots he got. I don't know what he pays for the robots. I think some of those robots, when I looked, brand new, they're $50,000, you know. I think he buys them on eBay and other places cheaper, but he got some money and it's an investment. So, the nanopore itself is this little, right there, that's the nanopore. That does all the rest of the DNA work after we do the PCR work. So I told you, we're sending Steve the tube of mushrooms. And that little tube I set him could have 96 mushrooms if I do 1 plate, or I could put up to 1,000, you know, from 1,000 tubes into that little tube. And Steve takes his pipette, puts it down in there, and, you know, the juice from 1,000 mushrooms. He takes 1 drop of it, and he puts it in that little nanopore. And that probably takes him 5 or 10 minutes. Has a computer on, his computer software that does the rest of the work. He can go then. He can go out to dinner, he can go on vacation, you know, go away for the day. Takes 24 hours for his computer to read everything in the nanopore to go through the cycle about. And that's how it's done. 24 hours later, he has all the raw data in his computer. He's not sitting there doing anything, does it automatically. So after he gets done with that, you know, he stores the mushrooms. I store mine in little plastic boxes and file cabinets. Steve taught me to store stuff in file cabinets. Now he stores them in other boxes. I can just keep mine in the file cabinet. This is the box Steve uses now. He uses these airtight boxes, so moisture doesn't get in to get your specimens to go bad. He doesn't use the plastic boxes on the inside of it. That's the exact box he's using. You get them at Home Depot. I don't know what they're about $20, I think. So after the nanopore is done, the next thing that comes is sequence validation. And so you get these A's, T's, and C's that come out of the machine, and you have to decide what it is. And you know, it's fun. If you want to take a class, maybe every month or 2, Fundus now has a volunteer sequence course. So you can sign up for this class. And it's 3 days long, 3 evenings long. You take it for like maybe 3 hours a night. And they teach you how to validate a sequence. It doesn't cost you any money. You're gonna get a free education. You can learn how it's done. You might not want to do it for them, but maybe you want to look at your own specimens. So you can take the class and they show you exactly what to do. And to become a sequence validator, you don't have to know anything, you don't have to have a science background, but you have to pass the test with 100% score. There is a test involved. And once you pass the test, you can work at stuff at any of the labs and help them. And it's not super hard.
I did it this winter and it was a wonderful experience. Very hands-on. They throw you right into the sequencing now.
Before you get to pay thousands of dollars for a college education to use this. You watch YouTube videos, you take all these free classes, you can be an expert at doing it. You're learning from all the experts. So skip that. So once you become a NAMA sequence validator, the validators, what they do is they get this little spreadsheet, they go online, and when the mushrooms come out of the app, where you might see the name of the mushroom, or this is the name the person thought the mushroom was. It could just say fungi because I didn't know what it was. This is a link to iNaturalist, it could be a mushroom observer. Little ID number, other names, the submitted ID. This is who's working on the mushroom. Like Kevin was working on these. Like if I'm doing the sheet, someone will have, somebody did it already, someone will be blank, so if it was me doing it, I type my name there. Then once you get it validated, you can put already validated, you can put there's an error, there's a problem, whatever. Then you put the name of the mushroom that you think it is. Once you validate it, you might put a little link there. We might, we have to answer questions like if it's a cryptic species that matches something. We check off that it's added to the mushroom observer, iNaturalist. We might put little notes like, this mushroom was the first ever to be found in Connecticut. We might make, this is the first mushroom ever found in North America. There's no matching sequences anyway. We could tell you all sorts of cool stuff about your mushroom that you found. We'll type that onto your iNaturalist account so you can read about it and really cool things that you find. Here's some of the comments. You would say it matches this. A lot of the comments we'll put would be validating. And the validators use this thing called MycoMap. And if you go to MycoMap, even if you just send your mushrooms in for one of the things, you can find your mushroom in a project. You won't be able to edit unless you take the class. Steve won't give you like editing. You can look at the raw data. You can change it if you took the class, but you can't if you haven't. But you can look at all your mushrooms, or you can look at mushrooms in your state on MycoMap. Here's some of my projects. If you go to MycoMap and look at your own records, you can find them there. You can click on the blast, which does a blast search. You can look at the sequence. You can do a lot of things. You can see if it was done or not. Oh, there it goes. You can even view the DNA and look for matches yourself there, if you have permission to do it. So I go on there, I say, do a blast search. And these things come up. What a sequence validator does is your mushroom usually is the top mushroom, and then it'll tell you what close match to it. If it says it's only a 97% match, that's not it. It's gotta be usually 99%, 99.5. You sort of have to look. It's not a set number, but you see like clusters, maybe everything from 99.9 to 99.7 is in one little cluster. That's one. If it's the next break in the line might be 92, then that's probably a different species. So you can't be that, but we try to match it. Program the validators use is called Eugene. It's a free program you can download off the internet. And that lets you look at the sequences, and you can, usually what you do is adjust an A or a T if something's off, you do some adjustment on it. I was told a paid fundus validator had to do 4 mushrooms an hour to get paid, I'm part of the volunteer validators, but they have paid ones in California. And I've recently told that there are 20,000 sequences currently that need validated. If a paid one has to do 4 an hour, if it's a volunteer, you might not be as good if you don't have to rush. How long would that take you to validate 20,000 sequences? And people wonder, why does it take so long why isn't my mushroom back tomorrow? There's a lot of stuff involved in this, and there was a lot of people helping. Before it used to be Steve and maybe a couple other people doing validating. I think they have 100 sequence validators now. These people aren't trained. These are just regular people like us. Come to Foray. We don't have a science background, and they're learning how to validate their sequences, how to do this. So it can be fun. It used to be like a fringe thing, but now it's becoming really, I mean, sequence validators everywhere I go. I've been to some Foray, says 5 people that are validating sequences. So on occasion, you might find an undescribed species. It gets named and published in a journal. So if you find the mushroom, you know, it doesn't end there, you know. It might say it's undescribed or make it a temporary code. And what happens is some of these people that describe mushrooms, like it might be Afraid of that works on Palladius, he might see your sequence on iNaturalist. You say, that's a new sequence, you know. He doesn't know what it is. He'll give, you know, send you an email or message out through iNat. He'll say, hey, I'm gonna describe this as new. And since you found it, you know, do you want to help me with the paper? I want to give you credit. And your name could be on that mushroom paper in a journal as helping with describing a new species. That happens often. You know, you get often asked to do that. Here's a new one that just got a name. It's just published. I just saw this on Facebook and find what's it results. Joshua just named this. It looks like a Fusiformis. It's a little bit thicker. If you want other resources, this is a, Steve has a lab video. So he had a class where you took it 2 days on Zoom and he recorded it, and you could pay to get access to the video. And he'll show you, like in the lab, like every step. You know, it might be 2 days worth of watching it, how he pipettes and how he does this. And he'll show you exactly and tell you exactly what to do. So you can run your own lab at home. It's not like you think, oh, this is complicated, I don't know what to do. His videos make it like simple. You just have to watch and do that little step and go to the next step. And it's not hard to do it anymore on your own. So other things that might frustrate your vulgar mind. So, you know, we used to be frustrated when we found that the dryad saddle polypoisclomosis changed to cereoporus. You know, we say, oh, 10 mushrooms that I knew changed names this year. That's frustrating. I'm really upset. It's much, much, much worse than that. You don't know how bad it's gonna get. It's gonna get worse, but it's also fun. So name changes. So Steve will tell you, and other people now, that 50% of the mushrooms in books are wrong. You know, it's not any, I'm not picking any author. Everybody does it 50% are wrong. You know, he'll tell you like Gallia rufa. There's more than one Gallia rufa. There's multiple ones. We have Gallia rufa INO1 and INO2. And these provisional names means it's probably not the real Gallia rufa, or they don't know what it is, but then there's more than one. And the IN means that it's first sequenced in Indiana. It could say PA01, it could say Connecticut 01. But that's what we're finding out. There's tons of cryptic species. Another thing Steve recently said is he's going to put a flag in there that somewhere the majority of names and past descriptions of North American macro fungi pre-2015 or so we ultimately found to be junior synonyms of something else that was previously described. 50%. 50%. And this is what's boggling my mind. Brian Hunt just posted this. There have been 1,930 provisional species names created on MycoMap from January 1st of this year to August 22nd of this year. So that's either stuff that we can't figure out what it is, or it could be something that the stream mushrooms did call to that, but it has a provisional name. A lot of this could be new. 1,930 mushrooms this year alone. I mean, how many of us know 1,930 mushrooms? And that's how many changes have been made. I mean, it's gonna get wild here in the future. I can imagine 10 years once the stuff starts getting figured out. You know, one mushroom, we thought it was one, there's 8 mushrooms that used to be Lactarius volimus. Now it's 8 mushrooms. And we don't have to learn how to tell them apart. Some will need DNA and some won't need DNA. We're sort of learning now. On a lot of them, we don't really need DNA. We can tell now that maybe it grown on a different tree type. You know, there's a lot of different things we're figuring out. You don't need DNA. You know, we needed DNA to find out at first, but now we don't need DNA to separate them. So, Devin also says, most species of mushrooms are misidentified in the historical and current literature. And he's talking about this. A lot of different changes. You know, I would say now that when I'm at Forays and, you know, I'm testing some stuff off the table, I might pick 20 mushrooms at a Foray to bring home and test. And I would say that 50% of the names I put on, and anybody else, me including, are wrong. When I get DNA back, it comes back as something else, or it comes back as something cryptic. 50% error rate. So we have a lot of work to do in mycology, and you can help. Get involved. It's simple. You don't have to open your own DNA lab. Just take part in the mycolist. Get 10 mushrooms for free, send them in, get them tested, and give it a try, and you'll be contributing to science. There's a very good chance that you will find new and undescribed species. I would say that of the mushrooms I send in, maybe 1 in 20 has not been, is undescribed. 1 in 20, high rate, high number. Anybody have any questions?
Sorry, I was late, so maybe you said at the beginning, so what doesn't get sequenced?
What?
What part of the genome?
ITS.
Sorry?
ITS region.
ITS.
Yeah, he's using ITS with the nanopore. There's other things can be done, but Steve only uses ITS for now, for the most stuff. That's what he does.
I think he said he puts stuff aside occasionally for when he expands into other areas.
Yeah, you might put chanterelles aside to do a different region or something else, and but 99% of the stuff that he puts on naturals or mushrooms are be ITS.
So what sort of spread in ITS percent ident-wise with those Galileo rufa taxa? Like would it be like a 2% spread or a 3%?
It could be off like it's 0.5% to be a different one.
A 0.5%?
It depends. You have to look at it. The chart, when you look at the graph and you'll see different clusters, there'll be a big break, like maybe a percent, half. You can just have to look where the break is. But definitely, if it's different a percent, it's something else. And it could be less than a break. It could be a 0.5% that separates something. But you can just notice there's a big, be a big cluster. You see 30 mushrooms in this one cluster. And all of a sudden there's a big break and there's another cluster down here. And that's, you sort of have to look at it individually to see where it breaks.
So it's not only how different the ITS sequences are, but where the differences are. And if it's
That's not where the difference are. It's like how they cluster up when you're comparing them.
Right, yeah.
But it's definitely if it's a percent difference, it's something else.
That's based on what I've learned, just in offhand ways.
But it could be a 0.5% too. It just depends.
Yeah, well, some genera too, like the ones that need like a different area of the genome, like in Morchella, if you do Morchella, you basically get lots of matches in the 99% that are just in the same section. So then you're gonna need another area of the genome, I guess. But like if you get a black Moreau, you'll get these matches in section distantes. But I guess if you have ones that cluster that are like the best ones, and they're a 0.5% better than the others, that's
And if you're doing your
enough
thing,
to say what the species.
you don't want to do a blast like on the regular blast site. You want to go to Mycomap and do your blast on Mycomap, because Mycomap's way more accurate. It'll way better. The other blast site won't probably help you a lot. You're going to do a Mycomap blast. Yes?
So to follow up on this question, if you're trying to figure out if it's a new species rather than let's like say a new variety, is it the same type of thing? Like you just look at how it clusters?
Yeah, we look how it clusters, and we know it's like something new. It's never been tested before because there'll be no matches. It'll show nothings. It'll be the only one. Like if I did a morale, it's something common. I might see 50,000 of that same, not 50,000, but hundreds and hundreds of them. Morchella escalenta, but if it's something new, there'll be nothing near it. Another thing we can do, another thing too, if you want to find out if your mushroom's rare or not rare, if you're using iNaturalist, I probably could do it on IMO, but iNaturalist for short, Steve will put a provisional name or whatever name he gives it on iNaturalist. You click on that and it'll say, show me other observations. So if I want to know if my mushroom came back, it's a name I never heard of before, I'll click it and I'll say, show me all the other observations. And it would say, oh, there's 100 of these or 500 of these. Or I could say there's only 3. And this is not scientific method, we're just guessing about this. But if it says there's 10 or less mushrooms that match it, you have a rare mushroom. there's been hundreds of thousands of things tested. It says there's 10 or less, it's rare. If you got 5 or less, it's rarely rare. So you can see how many other mushrooms match yours on iNaturalist just by clicking, show me other ones. If it says there's none, then you know you're really, really rare.
But some of those iNaturalist matches are not gonna be sequenced mushrooms, right?
Well, when you click the provisional name tag, it'll all be sequenced. Because if it has a provisional name, they've all been sequenced.
Oh, okay.
So it'll only be comparing it to sequence
Oh,
collections.
you have to, so you filter through provisional?
Yeah, like if yours came back as a provisional name, you just click it,
Wow.
show me all the other ones, and it's all been filtered because it's all been DNA tested.
Right,
You're not sorting through everything that was called,
yeah.
Mochella
because you-
americana.
Yeah, because you're gonna get a lot of observations that. would
Yeah, you're
not
searching the
be-
provisional name, and if it has a provisional name, it had to be DNA tested.
Oh, I see, okay.
Yes?
I have a question and a historical observation. First of all, I mean, I'm amazed at how far this all has come.
Okay.
And I was wondering who sells that sometimes unused lab equipment for so cheap?
Who sells it? I get it on eBay. Another place I've been finding.
No, I'm
What?
the people who sell it there. Where does this equipment come from?
Oh, like another place, where it comes from, another place I like to buy it. Okay, what's that place we went to? Relink Medical in Ohio. Like I go to that place. Yeah, I'm gonna tell you where they come from. But like I buy it at Relink Medical in Ohio. I said, where's it come from? And Relink Medical, I asked them, like, where do you get all these used medical cleanup equipment? And they do hospital cleanouts, lab cleanouts. So a hospital might decide they're gonna remodel and update everything. So they'll go to the hospital and they'll buy, or they might even buy, they might get paid to take it away. And they'll go to whatever hospital and they'll bring semi-trucks full of equipment back. It could be medical beds, it could be PCR machines, and there's disposal companies like that. And they'll get it and they'll put it up online for auction or.
Okay. So it's not always likely not the very, very latest equipment, but close.
It could be it could be 30-year-old equipment, or it could be a year-old equipment.
And the other observation I have, since you mentioned 30-year-old, this is actually more. I was in college in 1981, and I was in an evolution biology class, and I was supposed to give a talk about like genes and how they correspond, and they gave me proteins to do, which was from 61. I said, forget that. Got my hands on a Maxim and Gilbert paper that was 6 months old, and that was one of the essential early papers with fluorescent labeling about how to do DNA sequencing. So that was 1980 for historic perspective.
Okay.
Some of you may know that, but most of you probably not.
I know, like, one thing they teach you when you're learning is to run gel electrophoresis. Are you and these labs still doing that? I would imagine it'd be like
Hart told me that he does nanopore, he says he does gel.
Okay.
I do not do gel. When people did Sanger, they did this gel thing. And it cost about $1 a mushroom. You put your mushroom samples into this gel and it makes a band. If it has a band, it means it's successful DNA. If it doesn't have a band, it wasn't successful. And the reason why they did that in the past is because Sanger sequencing cost $6 a mushroom. And if you had a 50% failure rate, you know not to pay the $6 by testing these things. But it costs $1 a mushroom to do Sanger, to do gel, to do the gel. It's costing me $1 a mushroom to do nanopore if I do it the way I'm doing it now. So it costs me more money if I did the gel. Whether it failed or didn't fail, I'm not saving money. So I'm not doing gel anymore.
How concerning, I know Oxford Nanopore has come a long way since they came out, but they have serious error rates at the beginning. Has this been solved?
Yeah, I don't think they have too much problem. I think they're pretty accurate. Steve's telling me now, before he used to throw away mushrooms that had a Rick score of less than 10, but I'm hearing now that they're doing down way lower than 10 now. Like sometimes they're successful way lower.
Because in the field, Oxford Nanopore has been used very frequently to confirm sequencing when the accuracy is not too relevant. But when you want to know things on a qualitative level, but when a base per base accuracy is needed, I don't know if Oxford is there yet, also not.
They've been telling me now that it's just as accurate as Sanger, they've been comparing it. They've changed their chemistry, like, they update the Oxford, updates the chemistry and the way they're doing it, and it's improved, like, over the last number of years, it's improved greatly. Like, when it first came out, it wasn't so accurate, but now it's improved a lot.
Yeah, I had an anoscopy done by Steve and also by Linus Kuzma, who does Sanger, and the results were the same.
Identical, like, we've got identical results now. Yeah, I've been comparing stuff that's been done both ways, and it seems perfect either way.
I know as like a validator, we would be cleaning up a bunch of the like the nanopore sequences, but you know, it could still tell you what the specimen was.
Yeah, if we do Sanger, we have to edit that Sanger too. You have to edit it either way.
And I wasn't comparing with Sanger. Sanger is actually very old. I'm comparing with Illumina, with PacBio, so the true universal sequencing. And the reason why I found this concerning is because in the labs, and I happen to work in one that do human sequencing, also nanopore hasn't yet entered. So just a warning to the crowd here. So Oxford nanopore is being used, it has its value, and I do believe and trust that they're going to continue improving, but they haven't reached the level of accuracy and trustability to make it, to use it in humans, to use it for humans, and I think that is telling, because the price difference is incredible. So every big lab would jump immediately to also nanopore when they finally reach that level of accuracy. I just want to warn you, I'm with this, I'm enjoying this Foray a lot, but so that you know, Oxford nanopore has been battling for years for their place in the market, the place in the field, and I'd be super happy to hear that they reach the level of accuracy that Illumina and PacBio generate. Just a word.
Yes.
So I have a really lay person question.
Okay.
But I've read that in human genomic studies, they found that many genes stop between different chromosomes. And I was wondering how all this sequencing would reflect something like that. Would it see it as a different genus if a certain set sequence moved somewhere else?
I wouldn't know. It's too difficult for me. I don't have the background.
Okay. Thank you.
All right, well, thank you. If anybody has any other questions,